Basics of Medical Biochemistry. First edition, 2022. Chapter 11. Metabolism of Proteins and Amino Acids. Part 1 of 2. By Professor Dr. Muhammad Jawad. MBBS, Tao, M.Phil Philosophy, Ph.D., Dip M.E. Head of the Biochemistry Department-Fazaia Ruth Pfau Medical College-PAF Base Faisal-Karachi-Pakistan. Ex-Professor of Biochemistry, Qassim University Kingdom of Saudi Arabia. Ex-Professor, Head of the Biochemistry Department and Vice Principal. Liaquat College of Medicine and Dentistry and Fatima Jinnah Dental College, Karachi, Pakistan. Chapter 11. Metabolism of Proteins and Amino Acids. 11.1 Introduction. Nitrogen enters the body as dietary protein and leaves the body as urea, ammonia, creatinine, etc. In a 70 kg man, total body protein is about 12 kg with a relatively small amount of amino acid pool of 100 grams. 75% of amino acids obtained from the hydrolysis of body proteins are recaptured to form new proteins, whereas 25% serve as a precursor of other compounds. 1.1.1 Protein turnover. The rate of protein turnover is approximately 300 to 400 grams daily. The total amount of protein in the body remains constant, i.e., the rate of protein synthesis is equal to protein degradation. Turnover of digestive enzymes and plasma proteins is quite rapid, but the turnover of structural proteins is slow. As the half-lives of digestive enzymes and plasma proteins are very short, hours and days, but the half-lives of structural proteins like collagen and elastin are quite long, months and years. Overall protein turnover is maintained by following two factors. 1. Protein entry, by following routes. Dietary protein as a part of the food. Desquamation process, particularly on inner body surfaces. Synthesis of non-essential amino acids. 2. Protein exit, by following methods. Degradation, i.e., proteolysis. Catabolism of amino acids to urea, CO2 and H2O. Conversion of amino acid to glucose, ketone bodies, and fatty acids. Synthesis of purines, pyrimidines, porphyrins, creatine, etc. The primary role of protein is to provide amino acids for the synthesis of tissue proteins, and secondary as fuel, 1/5 of daily energy requirements. RDA for protein is 56 g/day for a 70 kg man, but the daily intake of protein is 70 to 100 grams in the typical American diet. A high-protein diet does not lead to the storage of amino acids, but a low-protein diet may cause serious nutritional problems like kwashiorkor and protein energy malnutrition. Figure 11.1 Protein intake and turnover. 1.1.2 Mechanism of protein degradation. It occurs by following two enzyme systems. 1. Ubiquitin proteasome system. This system operates in the cytosol and selectively degrades damaged or defective and short-lived proteins. It is an ADP-dependent process. Ubiquitin is a small protein that is found in almost all cellular tissues in humans and other eukaryotic organisms. Ubiquitin attaches defective proteins by isopeptide bonds. The energy for the formation of isopeptide bonds comes from ADP. The carboxyl group of terminal glycine residue covalently attaches epsilon amino groups of lysine on a protein designed to be destroyed. This is called ubiquitination of the target substrate, which is to be destroyed. Additional ubiquitin is then added to form a multi-ubiquitin chain. Polyubiquitinated proteins are recognized and degraded by a large barrel-shaped, multi-subunit protease complex, called the proteasome. The proteasome cuts the target protein into fragments, which are then further hydrolyzed by cytosolic proteases to amino acids. The ubiquitin is released and reused. The ubiquitin proteasome system utilizes the following three enzymes. E1, it is an activating enzyme that activates ubiquitin. E2, it is a conjugating enzyme that conjugates ubiquitin with the target protein and carried the ubiquitin target protein complex to E3. E3, it is a ligase that catalyzes the formation of isopeptide bonds between epsilon amino groups of lysine residues in substrate proteins and the activated carboxyl group of ubiquitin. Figure 11.2 Mechanism of protein degradation by the ubiquitin proteasome system. Protein degradation is influenced by some structural peculiarities of protein. These structural aspects are recognized as degradation signals. Proteins rich in proline, glutamate, serine, and threonine are rapidly degraded and they have short half-lives. The N-end rule indicates the destabilizing N-terminal amino acids such as arginine and acetylated alanine. But serine is a stabilizing amino acid. 2. Lysosomal degradation. Lysosomes secrete acid hydrolysis to non-selectively degrade the following categories of proteins. E1 intracellular proteins by autophagy. Extracellular proteins by heterophagy. 11.2 Sources and fate of amino acids. Amino acids are produced in the body as a result of the digestion of dietary protein, desquamation of body proteins and de novo synthesis of nutritionally non-essential amino acids. Extra amino acids are degraded immediately. Amino acids are catabolized in the following two phases. First phase, removal of the amino group as NH3 by transamination and deamination reactions. Second phase, C-skeleton of amino acids is metabolized in energy-producing metabolic pathways to produce CO2, H2O, glucose, fatty acids and ketone bodies, etc. Plasma amino acids are utilized in the synthesis of all tissue proteins, plasma proteins, enzymes, proteinous hormones, hemoglobin, antibodies, etc. They are also involved in the formation of non-protein nitrogenous compounds like epinephrine, choline, thyroid hormone, purines, pyrimidines, porphyrins, nicotinic acid, coenzymes, glutathione, nitric oxide, NO, histamine, serotonin, melanin, etc. Amino acids excreted through urine account for 1 gram per day. It represents 1 to 2% of urinary nitrogen. Amino acids present in urine are found both in free as well as in bound form, peptides and conjugates like hippuric acid. Most of the filtered amino acids are reabsorbed in proximal convoluted tubules. The reabsorptive capacity of the renal tubules is different for different amino acids. As daily urinary excretion of histidine is 188 mg in contrast to aspartic acid which is just 1.3 mg per day. The loss of amino acids in urine is proportionally increased with an increase in the plasma amino acid pool. It is seen in diabetes mellitus, acute infections, hepatic disorders and pregnancy. Amino aciduria occurs in many congenital defects in the metabolism of different amino acids. 11.3 Important reactions of amino acids. 1.3.1 Transamination. It is the transfer of the amino group, NH2, from an amino acid to a keto acid and the keto group from a keto acid to amino acid. As a result, amino acids become keto acid and keto acid become amino acids. Amino acid keto acid. Transaminase. Keto acid amino acid. Aminotransferase or transaminase moves the alpha amino group from an amino acid to alpha keto acid. These enzymes are found in the cytoplasm and mitochondria of almost all cells of the body. All amino acids, except lysine and threonine, are converted to glutamic acid, which then undergoes oxidative deamination and synthesis of non-essential amino acids. Features of transamination. It is a combination of deamination and amination. It is a reversible reaction. The main sites are the liver, kidney, heart, and brain but occur almost in all mammalian tissues. This reaction requires pyridoxal phosphate, vitamin B6, as a coenzyme. There are two clinically important aminotransferases i.e., alanine aminotransferase, ALT, or serum glutamic pyruvic transaminase, SGPT, and aspartate aminotransferase, AST, or serum glutamic oxalotic transaminase, SGOT. ALT transfers the amino group of alanine to ketoglutaric acid to form glutamic acid and alanine becomes pyruvic acid. AST transfers an amino group from glutamic acid to oxalotic acid to convert it into aspartic acid while glutamic acid is changed to ketoglutaric acid. Aspartic acid is then used in the synthesis of urea as a source of nitrogen. ALT or SGPT. Ketogluteric acid plus alanine glutamic acid plus pyruvic acid. AST or SGOT. Glutamic acid plus oxaloacetic acid, ketogluteric acid plus aspartic acid. Mechanism of transamination, ping-pong kinetic mechanism. All aminotransferases require the prosthetic group pyridoxal phosphate, PLP, which is derived from pyridoxine, vitamin B6. First step, the amino group of amino acid is transferred to pyridoxal phosphate, forming pyridoxamine phosphate and releasing keto acid. Second step, ketoglutarate reacts with pyridoxamine phosphate and produces glutamate. Figure 11.3 Ping-pong kinetic mechanism of transamination. Clinical significance of aminotransferases. Normal serum level of AST or SGOT is 0 to 35 U/L. It is raised in myocardial infarction, liver damage, but less than ALT, acute hemolytic anemia and muscular dystrophies. Normal serum level of ALT or SGPT is 7 to 41 U/L. It is raised in acute viral hepatitis, 250-1500 U/L. Both AST and ALT are raised in all hepatic disorders and they are useful in screening tests. 1.3.2 Deamination. It is the removal of an amino group from amino acid as NH3 with the formation of its corresponding keto acid. The site of occurrence of deamination is mainly in the liver and kidney. Types of deamination are oxidative deamination and non-oxidative deamination. Oxidative deamination, it is the removal of the NH2 group from an amino acid in the form of ammonia, NH3. The liberated ammonia is channeled into the urea cycle for detoxification or detoxication. This reaction takes place mainly in the liver and kidney. Oxidative deamination can occur in the following ways. A. By amino acid dehydrogenases. Glutamate dehydrogenase is the enzyme involved mostly. By this enzyme glutamic acid is converted to alpha-ketogluteric acid. Glutamic acid is the most abundant amino acid that undergoes this reaction. This reaction is also considered the chief source of NH3. The reaction produces NADPH2 from NADP, or NADH plus H plus from NAD+. This reaction is inhibited by GTP and ADP whereas activated by GDP and ADP. The direction of the reaction is decided by the relative concentration of substrates. In the post-absorptive phase, when glutamate levels in the liver are high, the reaction proceeds to form ammonia. In conditions of high ammonia levels, it drives toward the formation of glutamate. COOH HCNH2 glutamate dehydrogenase C equals O plus H2O plus NH3 CH2 CH2 NAD NADH plus H plus CH2 COOH glutamic acid ketogluteric acid. B. By amino acid oxidases. Oxidative deamination is also catalyzed by amino acid oxidases. The coenzyme is flavin mononucleotide, FMN. HO RCCOOH plus O2 plus H2 RCCOOH plus 2H2O2 plus NH3. Keto acid. NH2. Amino acid. Hydrogen peroxide is then split up by catalase. 2H2O2 2H2O plus O2. C. Bispecific amino acid oxidases, for example, glycine is deaminate by glycine oxidase to glyoxylic acid. H1/2O2 NH3 HCCOOH NH2O glycine glyoxylic acid. 2. Non-oxidative deamination. Serine and threonine, hydroxy amino acids, are deaminate by non-oxidative deamination. Enzymes are specific dehydrases and the coenzyme is pyridoxal phosphate. H. Serine dehydrase. CH2CCOOH CH3CO plus NH3. ONH2O. Serine pyruvic acid. 1.3.3 Deamidation. It is the removal of an amino group, amide group, from acid amide amino acids as NH3 by hydrolysis. Glutamine and asparagine are deaminate by glutaminase and asparaginase respectively. COOH. Asparaginase. HCNH2 plus H2O HCNH2 plus NH3. CH2CH2. NH2C equals OCOOH. Asparagine aspartic acid. 1.3.4 Decarboxylation. It is the reaction in which CO2 is removed from the COOH group of an amino acid by the enzyme decarboxylase. Coenzyme for this reaction is pyridoxal phosphate. The decarboxylation product of amino acid is an amine. The main sites of the decarboxylation reaction are the liver, kidney, brain, and intestine. R. Decarboxylase. CuCHNH2 RCH2NH2 plus CO2. Amino acid amine. Some of the amines are of great biological significance as mentioned in the table below. Table 11.1 Some important biological amines. Amino acid. Amine. Biological functions. Tyrosine. Tyramine. B.P. By vasoconstriction. Uterine contraction. Tryptophan. Tryptamine. B.P. By vasoconstriction. Tissue hormone. Serotonin. Histidine. Histamine. B.P. By vasodilation. HCl secretion. Pepsin secretion. Serine. Ethanolamine. Produce choline. Part of cephalin. Threonine. Propanolamine. Constituent of vitamin B12. Cysteine. Mercaptoethanolamine. Constituent of coenzyme A. Aspartic acid. Alanine. Constituent of pantothenic acid. Glutamic acid. Gamma-aminobutyric acid, GABA. Presynaptic inhibitor in brain. Lysine. Cadaverine. Product of putrefaction. Ornithine. Putrescine. Product of putrefaction. Arginine. Agmatine. Product of putrefaction. 1.3.5 Transamidation. It is the transfer of amide of glutamine to a keto acid or another substance. For example, glutamine transfers its amide group to a keto group of fructose 6-phosphate to form glucosamine 6-phosphate. The reaction is catalyzed by the enzyme transamidase or aminotransferase. Transamidase. Glutamine plus fructose 6-phosphate glucosamine 6-phosphate. The synthesis of asparagine is another example of transamidation. Asparagine is formed by the reaction of glutamine and aspartic acid. Asparagine synthetase. Glutamine plus aspartic acid plus ADP asparagine plus glutamic acid plus AMP plus pi. Mg2 plus 1.3.6 Transpeptidation. It is a chemical reaction in which an amino acid residue or a peptide residue is transferred from one amino compound to another. It is a reversible conversion of one peptide to another by a protease. The formation of glutathione is an example of a transpeptidation reaction. Glutathione is a tripeptide of 3 amino acids, gamma-glutamic acid, cysteine, and glycine. The synthesis of glutathione involves the following 2 steps. I. Formation of gamma-glutamal cysteine. It is formed by the condensation of gamma-carboxyl group of glutamate with the amino group of cysteine by the enzyme gamma-glutamal cysteine synthetase. Gamma-glutamic acid plus cysteine plus ADP gamma-glutamal cysteine plus ADP plus pi. 2. Formation of gamma-glutamal cysteine glycine. Glutathione. It is formed by the addition of glycine to gamma-glutamal cysteine by the enzyme glutathione synthetase. Gamma-glutamal cysteine plus glycine plus ADP gamma-glutamal cysteine glycine plus ADP plus pi. The formation of benzoyl glycine, hippuric acid, is another example of transpeptidation. This reaction takes place in the liver, where benzoic acid is detoxified by its conjugation with glycine to produce hippuric acid. The latter is then excreted in the urine. Clinical aspect. Penicillin, an antibiotic, irreversibly inhibits the enzyme transpeptidase by reacting with a serine residue of this enzyme. Penicillin binds at the active site of the transpeptidase enzyme that cross-links the peptidoglycan strands of bacteria. This reaction is irreversible and thus the growth of the bacterial cell wall is inhibited. 1.3.7 Transmethylation. It is a chemical reaction in which a methyl group is transferred from one compound to another. The methyl group containing amino acids such as methionine act as methyl donors. An example of transmethylation is the recovery of methionine from homocysteine.
Methionine synthase.
N5-methyl tetrahydrofolate plus homocysteine tetrahydrofolate plus methionine. Vitamin B12 as coenzyme. Another example of transmethylation is the formation of epinephrine from norepinephrine by the enzyme phenylethanelamine N-methyltransferase. Table 11.2 Classification of amino acids. Non-essential. Amino acids. Essential. Amino acids. Glucogenic. Amino acids. Ketogenic. Amino acids. Both glucogenic and ketogenic. Amino acids. Alanine. Arginine asterisk. Asparagine. Aspartate. Cysteine. Glutamate. Glutamine. Glycine. Histidine asterisk. Proline. Serine. Tyrosine. Lysine. Leucine. Isoleucine. Methionine. Phenylalanine. Tryptophan. Threonine. Valine. All non-essential amino acids, except for tyrosine. Plus, the following three essential amino acids: methionine, threonine, valine, leucine, lysine. Both are essential amino acids. Tyrosine plus the following three essential amino acids: phenylalanine, isoleucine, tryptophan. Arginine and histidine are considered essential amino acids in growing children. 11.4 Nitrogen balance. It is the quantitative difference between nitrogen input i.e., gain, and output i.e., loss from the body. Nitrogen intake. Most of the food nitrogen taken by humans is in protein form. When protein intake is 90 g/day, then nitrogen forms about 16% of protein, i.e., each gram of nitrogen comes from 6.25 grams of protein and the total nitrogen taken daily is 14.5 grams. Very small amounts of inorganic nitrogenous substances like nitrates and organic non-protein nitrogenous substances such as nucleic acids, phospholipids, and mucopolysaccharides are taken in food. There is also no storage form of protein in the body like that of carbohydrates, glycogen, or lipids, depot fat. Nitrogen output. Nitrogen is excreted or lost from the body through urine. This is the principal route of nitrogen excretion. The daily loss of nitrogen is 20 to 30 grams. It includes urea, uric acid, ammonia, creatinine, and creatine. Stools, feces, and sweat are other routes. Variable amounts of nitrogen are also lost in milk and menstrual blood in females in the form of exfoliated skin and mucosal cells and hairs. Following are the three states of balance between nitrogen input and output. 1. Nitrogen equilibrium. Normally an adult person under a normal adequate diet is in the state of nitrogen equilibrium, i.e., nitrogen intake equals nitrogen loss. 2. Positive nitrogen balance. When intake exceeds the output, it means that the body is in the state of protein anabolism and anabolism of other nitrogenous substances. It occurs in the following conditions: during growth, growing children, and pregnancy. Muscular exercise and training. Convalescence from states of negative nitrogen balance, example surgery and recovery from wasting illness. During the administration of anabolic hormones such as androgens, insulin, and growth hormone. 3. Negative nitrogen balance. it means that the output exceeds the intake and indicates excessive breakdown of protein, catabolism, and muscle wasting. It occurs in chronic depleting diseases such as diabetes mellitus, Cushing's syndrome and hyperthyroidism. Chronic illnesses, example chronic renal, cardiac, gastrointestinal disorders, hepatic and pulmonary diseases, infections, and fevers. Wasting diseases such as AIDS, cancer, and tuberculosis. Inadequate dietary intake of proteins, as in, starvation and deficiency of one or more of the essential amino acids. Malnutrition, example protein and energy deficiency syndromes such as kwashiorkor and marasmus. Loss of proteins, albuminuria or proteinuria, as in, chronic hemorrhage, extensive burns and trauma. Lactation with an inadequate diet. Increased secretion or administration of thyroid hormone, ACTH, and glucocorticoids. 11.5 Formation, transport, and fate of ammonia. A significant amount of ammonia is produced in different body tissues from the catabolism of amino acids by deamination and deamidation reactions. A considerable quantity of ammonia is also produced in the gut by intestinal bacterial flora, from dietary proteins, from urea present in fluids secreted into the gut, from a means of diet as well as obtained from hormones or neurotransmitters by the action of monoamine oxidase, from the catabolism of purines and pyrimidines. The transport of ammonia from peripheral tissues to the liver takes place by following two mechanisms. 1. By the formation of glutamine, ammonia combines with glutamate to form glutamine by the enzyme glutamine synthetase. Glutamine synthetase. Glutamate plus NH3 plus ADP glutamine plus H2O plus ADP plus pi. Glutamine is considered a non-toxic form of ammonia and is transported to the liver, where it is hydrolyzed by glutaminase to give NH3 and glutamate. Glutaminase. Glutamine plus H2O glutamate plus NH3. Glutamate undergoes oxidative deamination to give alpha-ketoglutarate and ammonia by the enzyme glutamate dehydrogenase. 2. By glucosalanine cycle, in peripheral tissues, muscle, alanine is formed by the transamination of pyruvate. The pyruvate is formed by aerobic glycolysis as well as from the catabolism of branched-chain amino acids, isoleucine and valine. In the liver, alanine is transaminated with ketoglutarate by ALT to give pyruvate and glutamate. Pyruvate is converted to glucose and transported to muscles. Glutamate is deaminated by glutamate dehydrogenase to produce ammonia. Figure 11.4 Transport of ammonia from muscle to the liver showing glucosalanine cycle. The ammonia is absorbed into the portal circulation, taken up by the liver and is finally detoxified into urea. Under normal conditions of health, the liver is quite able to remove all ammonia from the blood. The clinical significance of this fact is that even a small quantity of ammonia in blood is toxic to CNS and it is one of the causes of hepatic encephalopathy. About 80 to 90% of the total ammonia produced is shunted into the urea cycle, with the remaining 10 to 20% metabolized by peripheral tissues, including the kidney, heart, and brain. Normal blood ammonia level is 18 to 60 μg/dl. The concentration of ammonia in portal blood is relatively high as compared to systemic blood. The portal blood may bypass the liver in severe hepatic disorders such as cirrhosis. The portocaval shunts produced surgically are also conducive to ammonia intoxication. Ammonia intoxication is exaggerated after ingestion of large amounts of proteins or after hemorrhages into the gut. 1.5.1 Hyperammonemia. It is a metabolic disturbance characterized by an excess of ammonia in the blood. It is a dangerous condition that may lead to encephalopathy and death. It may be primary, inherited, or secondary. Primary hyperammonemia is caused by several inborn errors of metabolism that are characterized by reduced activity of any of the enzymes in the urea cycle. Secondary hyperammonemia is caused by inborn errors of intermediary metabolism characterized by reduced activity in enzymes that are not part of the urea cycle, example propionic acidemia, methylmalonic acidemia. Acquired hyperammonemia occurs in cirrhosis of the liver, due to the formation of collateral circulations and when urea synthesis is decreased. The toxicity of high levels of ammonia is thought to result from a shift in the equilibrium of the glutamate dehydrogenase reaction toward the direction of glutamate formation. Hyperammonemia depletes ketoglutarate from the brain. This leads to inhibition of the TCA cycle, which is the main source of energy in the brain. So, the brain is deprived of its main source of energy, leading to encephalopathy. Glutamate dehydrogenase. Ketoglutarate plus NADP plus H plus NH3 glutamate plus NADP plus. Features of ammonia intoxication are flapping tremors, slurring of speech, vomiting, blurring of vision, coma, and finally death if the condition is not properly treated. Treatment includes restriction of dietary proteins and administration of compounds like phenylbutyrate to increase the excretion of non-essential amino acids, after converting them into nitrogen-containing molecules. Figure 11.5 Metabolism of ammonia. 11.6 Urea cycle. The metabolic pathway that synthesizes urea is known as the urea cycle, which is composed of several enzymes acting in sequence. It is also known as the Krebs-Henselite cycle and the ornithine cycle. Source of urea. Ammonia results from the deamination of amino acids. From salt like ammonium carbonate and ammonium lactate. From amino acid arginine, which split up into urea and ornithine. There are two nitrogen atoms in the urea molecule, one is supplied by NH3 and another by aspartate. The carbon and oxygen of urea come from CO2. Site of urea cycle. Liver is the main organ where the urea cycle takes place. In the brain and mammary gland to some extent, and kidney up to arginine. The first two steps of the urea cycle occur in the mitochondria and the last three steps in the cytosol. Biological importance of the urea cycle. The major biological role of this pathway is the detoxification of ammonia. Ammonia is converted to the less toxic substance urea before excretion in urine by the kidneys. The urea cycle also serves for the biosynthesis of arginine from ornithine in the liver, kidney, and intestinal mucosa. Kidney and intestinal mucosa probably contribute most of the body's arginine because they possess all the urea cycle enzymes except arginase. Hence they can form up to arginine and cannot form urea. Arginine is used for protein synthesis. The urea cycle may also be connected to the TCA cycle and produce energy. Amount and blood levels of urea. Normal blood urea level is 20-40 mg/dl. Daily production of urea is about 30 g. About 80 to 90% of urinary nitrogen is due to urea, the others are creatinine 5%, ammonium ions 3% and the remaining 12%. Urea is routinely measured in the blood as blood urea nitrogen, BUN. It is half of blood urea level, i.e., 10-20 mg/dl. The levels may be raised, uremia, in both acute and chronic renal failure, urinary tract obstruction, and congestive heart failure. Urea excretion is decreased during starvation. 1.6.1 Steps and reactions of the urea cycle. The urea cycle includes the following 5 enzymatic reactions. The first 2 steps take place in mitochondria and the other 3 in the cytoplasm. 1. Formation of carbamoyl phosphate. The formation of carbamoyl phosphate takes place by the combination of NH3 and CO2 as HCO3 using 2-ADP. Ammonia provides the first nitrogen atom of urea. This reaction is catalyzed by mitochondrial carbamoyl phosphate synthetase I, CPSI. It is a rate-limiting enzyme. It needs biotin as a coenzyme, Mg2+ and N-acetyl glutamate as an activator. Note: CPS2 is used in the biosynthesis of pyrimidines. 2. Formation of citrulline. Carbamoyl phosphate reacts with ornithine to form citrulline. This reaction is catalyzed by ornithine transcarbamylase, OTC. Citrulline in turn inhibits OTC activity. 3. Formation of argininosuccinate. The citrulline, produced in mitochondria, is transported to the cytosol by antiporter in exchange for ornithine. Citrulline reacts with aspartate to form argininosuccinate. Aspartate provides a second nitrogen atom of urea. This reaction is catalyzed by argininosuccinate synthetase and requires 1-ADP. It is inhibited by arginine, AMP, and argininosuccinate. For formation of arginine, argininosuccinate is cleaved in by argininosuccinase to form fumarate and arginine. Argininosuccinase is inhibited by fumarate and arginine. Arginine acts as the immediate precursor of urea. The fumarate provides a link to the TCA cycle through malate. This relationship between the urea cycle and the TCA cycle is named as Krebs bicycle. 5. Formation of urea and ornithine. In the cytoplasm, arginine is cleaved by arginase to give urea and ornithine. The regeneration of ornithine in each turn of the urea cycle is just like the regeneration of oxalostate in the TCA cycle. It is transported to mitochondria to react with carbamoyl phosphate and thus repeats the cycle. Figure 11.6 Reactions of REA cycle. Overall stoichiometry of urea cycle. NH3 plus HCO3 plus H2O plus 3 ADP plus aspartate. Urea plus 2 ADP plus AMP plus 2 pi plus PPI plus fumarate. 1.6.2 Fate of urea. Urea produced by the liver is released into the bloodstream where it travels to the kidneys and then ultimately excreted in the urine. A small amount of urea is diffused to the intestine where it is cleaved to CO2 and NH3 by bacterial urease. This ammonia is partly lost in the feces and partly reabsorbed into the blood. In cases of renal failure, plasma urea level is elevated, stimulating a greater transfer of urea from the blood into the gut. The ammonia produced in the gut by the action of urease on urea becomes a clinically important source of ammonia, causing hyperammonemia frequently seen in these patients. Figure 11.7 Krebs bicycle, the connection between urea cycle and TCA cycle. 1.6.3 Regulation of the urea cycle. It occurs principally at CPSI. N-acetyl glutamate is an allosteric activator of CPSI. N-acetyl glutamate is synthesized from acetyl-CoA and glutamate by N-acetyl glutamate synthase. This enzyme is activated by arginine. Enzymes of the urea cycle are controlled, long-term regulation, at the gene level with long-term changes in the quantity of dietary protein. A long-term increase in the number of dietary proteins leads to a significant increase in the concentration of the enzymes of the urea cycle. The cycle is also regulated by the availability of the respective substrate as short-term regulation. 1.6.4 Disorders of the urea cycle. Most of these disorders occur due to a deficiency of enzymes taking part in the urea cycle. 1. Argininemia. It is an autosomal recessive urea cycle disorder where a deficiency of the enzyme arginase causes a buildup of arginine and ammonia in the blood. The arginase catalyzes the hydrolysis of arginine to produce ornithine and urea. Although customarily considered in terms of its role as the final enzyme of the urea cycle, the enzyme is also found in a variety of non-hepatic tissues. Mental retardation is the main clinical feature of argininemia. This condition is treated by an arginine-free diet like nuts and seeds, meat products, legumes, seaweed, etc. 2. Argininosuccinic acidemia. It occurs due to the deficiency of argininosuccinase. Excess ammonia in the blood can cause brain damage manifested by vomiting and convulsions. 3. Hyperammonemia type I. It is due to a defect in CPSI. 4. Hyperammonemia type 2. It is due to a defect in OTC. It is the commonest urea cycle disorder. 5. N-acetyl glutamate synthase deficiency. It causes the deactivation of CPSI. 6. Ornithine translocase deficiency. It results in the failure of transport of ornithine from the cytosol into the mitochondria. 11.7 Metabolism of individual amino acids. 1.7.1 Metabolism of glycine. It is a neutral, aliphatic, glucogenic, and non-essential amino acid. It can be produced from serine, threonine, and glyoxylic acid. Synthesis of glycine from serine. Glycine is obtained from serine by a reversible reaction in the presence of tetrahydrofolate, which is converted to N5N10-methylene-tetrahydrofolate. The enzyme for this reaction is serine hydroxymethyltransferase. This enzyme needs pyridoxal phosphate, PLP, as a coenzyme. Figure 11.8 Synthesis of glycine from serine. Synthesis of glycine from threonine. It occurs as follows. Figure 11.9 Synthesis of glycine from threonine. Synthesis of glycine from glyoxylic acid. The transamination reaction catalyzed by glycine transaminase in the cytosol of the liver is a salvage process for regenerating glycine at the expense of glutamate or alanine. Figure 11.10 Synthesis of glycine from glyoxylic acid. Glycine is oxidized to NH3 and CO2 by of glycine cleavage system, a liver mitochondrial enzyme. Clinical aspect: failure of this salvage process due to the deficiency of glycine transaminase delivers glyoxylate into oxalate stone formation, leading to kidney damage and resulting in primary hyperoxaluria. Some other metabolic roles of glycine are: it is a part of glutathione, which is formed of 3 amino acids, glutamic acid, cysteine, and glycine. It is formed in the cytoplasm of the liver with the help of ADP and gamma-glutamyl-cysteine synthetase and glutathione synthetase. Glutathione exists in 2 states: reduced form with a free SH group, GSH, and oxidized form without a free SH group but a disulfide linkage, GSSG. It helps in the absorption of amino acids and protects the hemolysis of RBCs by breaking H2O2, which causes cell damage. Like other amino acids, it is involved in the synthesis of many proteins. It is required for the synthesis of purines. Carbons 4 and 5 and nitrogen 7 of purines are derived from glycine. It can be converted to serine. It is involved in detoxification processes by conjugation reactions. It is conjugated in the liver with benzoic acid, a toxic food additive, giving hippuric acid, which is excreted in the urine. Glycine conjugation is used to detoxify several other drugs. Figure 11.11 Conjugation of glycine with benzoic acid to produce hippuric acid. It also produces primary bile acids, i.e., glycocholic acid and glycoanodioxycholic acid after conjugation with cholic acid and chinodioxycholic acid respectively in the liver. It acts as an inhibitory neurotransmitter in the central nervous system. It participates in the synthesis of creatine. Creatine, creatine phosphate, phosphocreatine, and creatinine. Creatine is a non-protein nitrogenous compound. Creatine is naturally synthesized from 3 amino acids, glycine, arginine, and methionine. It is synthesized primarily in the kidney and liver and then transported to the blood for use by muscles. Creatine phosphate, the phosphorylated derivative of creatine, is found in muscles. It is a high-energy compound that can reversibly donate a phosphate group to ADP to form ADP. The reaction, catalyzed by creatine kinase, CK, provides a small but rapidly mobilized reserve of high-energy phosphates that can be used to maintain the intracellular level of ADP during the first few minutes of intense muscular contraction. Creatine and creatine phosphate spontaneously cyclize at a slow but constant rate to form creatinine, which is excreted in the urine. Clinical aspect: the amount of creatinine excreted from the body is proportional to the total creatine/creatine phosphate content of the body, and thus can be used to estimate muscle mass. When muscle mass decreases for any reason, example in the case of muscular dystrophy, the creatinine content of the urine falls. Similarly, any rise in blood creatinine is a sensitive indicator of kidney failure, because creatinine is normally rapidly removed from the blood and excreted. The daily excretion of creatinine in a typical adult man is approximately 1 to 2 g. Metabolic disorders of glycine. Primary hyperoxaluria is due to failure of catabolism and or transamination of glyoxylate either to formate or glycine, respectively. The accumulated glyoxylate is oxidized to oxalate, which causes bilateral calcium oxalate stones, nephrocalcinosis, recurrent urinary infection, and early mortality from renal failure or hypertension. Glycinuria is due to defective renal reabsorption of glycine, leading to excretion of 0.6 to 1.0 g glycine per day with normal blood glycine level. The patient tends to precipitate renal oxalate stones. Figure 11.12 Creatine, creatine phosphate, and creatinine. 11.7.2 Metabolism of alanine. Alanine is a neutral amino acid with a nonpolar side chain. It is a non-essential amino acid and is synthesized by following a reversible reaction of transamination by the enzyme alanine aminotransferase, ALT, or serum glutamic pyruvic transaminase, SGPT. ALT or SGPT. Glutamic acid plus pyruvic acid ketoglutaric acid plus alanine. The alanine is produced in muscle by transamination reaction, then carried to the liver where it is converted back into pyruvate by the same enzyme and the same reaction of transamination. The pyruvate can be used for gluconeogenesis and glutamic acid provides ammonia for urea synthesis. This transport is referred to as the glucosalanine cycle. 11.7.3 Metabolism of serine. It is a type of neutral, non-essential and hydroxy amino acid. It is synthesized reversibly from glycine. Serine can also be derived from 3-phosphoglycerate, first by its oxidation to 3-phosphopyruvate and then transamination to 3-phosphoserine. The hydrolysis of phosphate ester produces serine. Some important metabolic roles of serine are: D-serine may act as a neurotransmitter in the brain. It participates in making beta-pleated sheet structure of protein along with glycine and alanine. It acts as a carrier of phosphoric acid in phosphor protein, due to the presence of the O group. It is utilized in the synthesis of purines, pyrimidines, cysteine, choline, and cephalin. It can give pyruvic acid by the enzyme serine dehydratase. Serine plus H2O pyruvic acid plus NH3 plus H2O. 11.7.4 Metabolism of threonine. It is a neutral, hydroxyl, and essential amino acid. Important metabolic roles of threonine are: it is dehydrated to ketobutyrate, then to propionyl-CoA and then finally to succinyl-CoA, which enters the TCA cycle. It can give glycine by threonine aldolase. 11.7.5 Metabolism of glutamic acid. It is an acidic amino acid, produced in the cytosol of liver cells by the transfer of an amino group to ketoglutaric acid. Glutamic acid enters the mitochondria and undergoes deamination to give ammonia, which is then converted to urea by the urea cycle. Other important metabolic roles of glutamic acid are: it gives rise to glutathione in combination with cysteine and glycine. It combines with ammonia to form glutamine by the enzyme glutamine synthetase. Glutamine is considered a non-toxic form of ammonia and is transported to the liver, where it is hydrolyzed by glutaminase to give NH3 and glutamate. A neurotransmitter, gamma-aminobutyric acid, GABA, is produced by the decarboxylation of glutamic acid. The action of GABA is postsynaptic inhibition in CNS. By transamination, it is converted to ketoglutaric acid, which can enter the TCA cycle. 11.7.6 Metabolism of glutamine. It is an amide derivative of glutamate in which the side chain COOH group is amidated. Synthesis. It is formed by the combination of ammonia with glutamate by the enzyme glutamine synthetase. Glutamine synthetase. Glutamate plus NH3 plus ADP, glutamine plus H2O plus ADP plus pi. Fate. It is hydrolyzed by glutaminase to give NH3 and glutamate. The NH3 so produced is used to neutralize H+ in renal tubules. Glutaminase. Glutamine plus H2O, glutamate plus NH3. It acts as a carrier of NH3 from various tissues of the body to the liver, where NH3 is detoxified to urea. The formation of glutamine in the brain prevents the accumulation of NH3. 11.7.7 Metabolism of aspartic acid. It is an acidic and non-essential amino acid. Synthesis. It is synthesized in the body by a transamination reaction between glutamic acid and oxalotic acid. AST or SGOT. Glutamic acid plus oxalotic acid, ketoglutaric acid plus aspartic acid. Fate. By the reverse of the above reaction, it is converted to oxalotic acid. Oxalic acid enters the TCA cycle for oxidation or undergoes gluconeogenesis to form glucose. It is used in the synthesis of urea as a source of nitrogen. It is used in the synthesis of purines and pyrimidines. It can be converted to asparagine, which is formed by the reaction of glutamine and aspartic acid. Asparagine synthetase. Glutamine plus aspartic acid plus ADP, asparagine plus glutamic acid plus AMP plus pi. Mg2+. Aspartic acid also acts as an excitatory neurotransmitter in CNS. 1.7.8 Metabolism of asparagine. It is an amide derivative of aspartate in which the side chain COOH group is amidated. Synthesis. Asparagine is formed by the reaction of glutamine and aspartic acid by the enzyme asparagine synthetase in the presence of Mg2+ ions. This reaction is an example of transamidation. Glutamine plus aspartic acid plus ADP asparagine plus glutamic acid plus AMP plus pi. Fate. It is hydrolyzed by asparaginase to give NH3 and aspartate. The NH3 so produced is used to neutralize H+ in renal tubules. Asparagine plus H2O aspartate plus NH3. Aspartate obtained from asparagine can be converted to oxaloacetic acid which enters the TCA cycle, while NH3 is detoxified to urea. Exercise questions. For MCQ 6 to 10 see Part 2 of this chapter. MCQ 1. Which of the following amino acids in the liver is incorporated to nitrogen that is derived from the breakdown of amino acids in skeletal muscles? Alanine, aspartate, glutamate, glycine, phenylalanine. MCQ 2. Which of the following compounds in acidosis is diverted from the liver to the kidney to raise the body's pH? Ammonia, asparagine, hydrogen ions, glutamine, urea. MCQ 3. Which of the following amino acids has abnormal metabolism in albinism? Alanine, phenylalanine, tryptophan, tyrosine. MCQ 4. A 3-day-old neonate presented with seizures due to a suspected urea cycle defect. Lab reports revealed raised levels of serum ammonium ions, glutamate, and alanine with normal serum citrulline and normal level of urinary orotate. Which of the following enzymes of the urea cycle is defective in this patient? Arginase, argininosuccinase, argininosuccinate synthetase, carbamoyl phosphate synthetase I, ornithine transcarbamylase. MCQ 5. Which of the following compounds is also produced in the reaction of the urea cycle shown below? NH3 plus CO2 plus aspartate rightward arrow urea plus, citrate, fumarate, glutamate, lactate, pyruvate. Please write answers in the comments or contact drmjot@yahoo.com. End of Part 1 of 2 of Chapter 11. Thank you.